The overall goal of this procedure is to demonstrate the interaction between the antiviral Mx1 protein and its viral target, the influenza nucleoprotein, via co-immunoprecipitation. This is accomplished by first lysing transfected cells containing both proteins in the presence of N-ethylmaleimide. The second step is to add a nucleoprotein-specific antibody to bind to the influenza nucleoprotein and the interacting Mx1 protein. Next, the resulting immune complexes are precipitated with protein G beads, and the contaminating proteins are removed through extensive washes. The final step is to elute the influenza nucleoprotein and the co-immunoprecipitated Mx1 protein from the protein G beads. Ultimately, Western blotting is used to show the interaction between the influenza nucleoprotein and the antiviral Mx1 protein.
The main advantage of this technique over existing methods like yeast to hybrid is that the interacting proteins are studied in their natural environment, which is the human cell. This method can provide insight into the interaction between the influenza A virus nucleoprotein and the antiviral Mx1 protein, but it can also be modified to study other weak or transient protein-protein interactions. The technique will be demonstrated by Dorien de Vlieger, a colleague from my laboratory.
Prepare all of the transfection buffers as described in the accompanying text protocol. Dilute the plasmid DNA into 600 microliters of tris EDTA. Then, add 150 microliters of Heaps buffered calcium chloride in a dropwise manner to the plasmid samples, and mix by pipetting up and down 3 times. Add the resultant 750 microliters of plasmid solution dropwise to 700